Hello, everyone. Um, Welcome to our webinar series. This is a third webinar of technical in nature. We are very much pleased to introduce you to our new product launch, Synomics Discover. Just before we start the presentation, I want to give you a bit of a background about Synomics as a company. Synomics is a genome discovery, multi-omics discovery company, as well as a genomic trait improvement company. Is one of the portfolio company of the Groviner Food and AgTech, previously known as the Wheatsheaf Group. It's an international investor in food and agriculture business. So what does Synomics do is to provide novel, actionable, biologically relevant insights to help our partners to unlock those insights much better than what you can achieve with the current state-of-the-art tools or best-in-class tools. And we also help our customers and partners to translate those novel insights and features to trait improvement and genetic improvement programs. So we have put together today an agenda that consists of 3 parts and involves 3 presenters, starting with me. And towards the end of this presentation, you will have a short video followed by a Q&A session. So please do stick around to remain in the last interesting part of the webinar. So moving on to part 1, so that's for me. I'm going to talk about why Synomics is needed. And before I start to give that talk, I want to introduce myself. My name is Haja Kedar-Medin. I'm a Chief Technology and Science Officer responsible for research, innovation, and technology platform development, as well as looking after the product development of our companies such as Synomics Discover. I head up a highly talented R&D team across technology teams across 3 countries, UK, US, and Denmark. Background, veterinarian until 1990s, early 90s. From then on, 30 years of professional career as an animal geneticist, breeder, data scientist, bioinformatician, and working in also computational systems biology, primarily in animals, but also in humans and recently in crops. So these experiences were across academia, private sector, as well as the federal and state government organizations. To be precise, 6 countries and 4 continents. So that's me. And moving on, I want to start off with a slight primer to this discussion, a primer to the Discover presentation on biological data collection and what we have been witnessing in terms of the biological data, starting with genomic data. There has been a rapid evolution of the volume, the shape, the type of biological data that we have been collecting over the last 20 years. And to give a historical perspective about genomics in general, but multi-omics in specifically, I have separated this into sort of a 20-year block, anything after 2000 and pre-2000. What happened in pre-2000, and we call it as a pre-genomics era, is that, for instance, if we take a livestock or pet or companion animals, we had a pedigree recording, phenotype performance, disease recording. We did have genetic markers, a small number in hundreds and thousands, few thousands of markers such as RFLPs, microsatellites, etc., that was then used to sort of map to the genome and the phenotype bank being mapped to those markers in order to identify potential causative variant that is in linkage to Ziglurum with those markers. The idea was to be able to utilize that information to identify regions of the genome, so-called QTLs, and use that in a marker-assisted selection or integration program. So that was pre-2000. And what happened in mid-2000s and early 2000, we call it as the early genomics era, that's when we moved into a slightly higher number of genomic profiling as in high-density SNP genotyping, for instance, where we moved up to a few thousands of SNPs per individual livestock, animals, pets, and crops, and so forth. Genomic profiling then was expensive. It was often limited to experimental conditions, and few industries were practicing that, actually to implementing that. That was because it was expensive, it was questionable. We were still developing statistical genetic, quantitative genetic models to accommodate the wealth of genomic information that was coming in. Computationally, it was difficult too, because to store a large amount of data and process that, transfer that, etc. Then came 2010s, and that was an era where we call it the genome sequence era, where we were moving from first generation, second generation, third generation sequencing, what we call next generation sequencing. The cost came down quite rapidly and dramatically from what you call as a $1,000 genome to now you can get sequenced for $100 to $200 per individual. It is also the time the other multi-omics data started coming in, for instance, transcriptomics, epigenomics, and etc. And 2010s, we are really looking at not just high volume of whole genome sequence and exome sequence data, but also RNA-seq, bulk RNA-seq, single-cell RNA-seq, epigenome, histone modification, methylation capture, large-scale protein and mass spec assays, etc., etc. So the key message here from this slide is that we are dealing with mountains of data, multi-omics data, and it is actually cost us so low that we do not see a problem to create mountains of data and also to be able to store and process the data computationally. But is that tremendous improvement in biological data collection, various types? Did it actually help to understand the complex biology behind phenotypic differences in animals and plants, disease propensity, etc.? Our understanding of disease, performance, and resilience, or sustainable characteristics, has not improved as we once hoped. I'll give you a little bit of rationale for that, but I want to quote one thing from Francis Collins, who was the head of the Human Genome Project back in 2000. He said, in the next 15 to 20 years, you will see a complete transformation of therapeutic medicine. But actually, what eventually happened was, even those high-profile journals like Nature, JAMA, and so forth, which published papers like there are a few genes that controls complex human disease, for instance, psychiatric diseases, so schizophrenia, bipolar, and other behavioral disease, they needed to be retracted because what became clear afterwards is highly polygenic, multiple etiological factors, genetic factors, and complex biology behind this disease. And it kind of reflects back on other species in animals and pets. We are dealing with a complex trait architecture, disease biology. And part of that is because we are no longer dealing with a single gene or a few gene or independent effects. We are looking at complex interactions between different types of data. So if you think about genomic data, you can have interactions between genes, gene-gene interactions, variant-to-variant interactions. Epistasis is one clear example of trait architecture. If you move up, protein-protein interaction, gene-protein interactions, how the microRNA controls the mRNA abundance, how does the epigenetics come into play with gene expression, and then top on that, microbiome. So there is a huge amount of type of complex interactions that happens in the genome in life science areas that is difficult to really understand the causation and regulatory genomics of these diseases. So the question is, what if we can understand more of the complex biology and genomic architecture behind this kind of important phenotypes and diseases? What if we could explain more clearly the features, the networks that are behind this complex architecture? And if we do know that, what we could do something about it. So with that introduction, I want to say, so are we ready to sort of tackle those challenges? There are key challenges in this multi-omics discovery, starting with genome discovery. and also utilizing other types of data. There are three pillars of those challenges. One is computational, next is statistical, the other one is biological. Computationally, high dimensionality, huge dimensionality, both the shape, size, and types of data. You could, for instance, in dairy cattle or in cattle, with low-pass sequence data, we can get 60, 70 million SNPs on tens of thousands of individuals. It costs about the same to generate that type of data as we did with small density or high density SNP chips. You can also generate other multi-omics data in very large sample size. So the ability to store and process the data is still a challenge. Statistically, there's no standard statistics that can deal with this multi-omic, multi-million combinations of data points. Test statistics, test parameters. The key problem or challenge that rather is the redundancy in these kind of high dimensional features. It is difficult to sort of pinpoint and increase the signal to noise ratio, to have a better signal to noise ratio here. Or also, how do we actually rank important features that we can then take it further to biological, biotechnological, biopharmaceutical, biomedical interventions, as in drug targets and biomarkers, and eventually to apply that in real practice. So that is a key challenge. So it is clear that while we have all of these developments in data generation, including phenotypic data, different types of phenotype data, the challenges remain, and really we need to think about different way of approaching this issue. And here is the prelude to what's going to come after my presentation. And we're going to focus on really capturing those combinations or interactions between and within different biological data. So when you have, what you have on the left-hand side is an illustration of what the multi-omics data look like and the interactions. On the right-hand side, we are kind of comparing and contrasting what does our method do in relation to standard best-in-class tools, discovery tools, as well as deep learning and machine learning tools. So the first on the left-hand side, what you see here is a genomic layer, and then you can have another omics layer, which could be another half a dozen of those, starting with transcriptomics, epigenomics, metagenomics, and so forth. For each individual layer, you have not only single feature impact on the phenotype of disease, it is a complex interactions between them. So you can capture that with our analytical platform. But more so that those kind of networks, gene networks, or interacting SNPs, etc., how does that translate to other multiple biological layers? So the analytical engine that we built in is based on combinatorial analytics. It's a set of constraint-based models applying Boolean algebra, which for some of you, it is about conjunction, elimination, and a lot of logic. So it's really biologically and mathematically constructed in a way that it makes sense rather than blindly predicting an outcome without biological relevance underneath those prediction models. And that's what happens with the AI and machine learning. We got an input layer, an output layer. There's a bunch of hidden layers where we don't really control the biology, but whereas we can in terms of the interpretations of those combinations in a biological context. So with that introduction to why do we need a tool of that type and size, given these improvements, I will hand this over to my colleagues, Tobias and Ryan, who will then take you through what is Synomics Discover, how it works, and what are the key applications in target species of agricultural importance. So with that, I hand this over to Tobias.
Thank you, Haja. So my name is Tobias Røykjær. I'm a senior data scientist here at Synomics. I have a background in computer science, data science, and bioinformatics. And I will, together with Ryan Morrison here today, present some of the technical details of the Synomics Discover pipeline. So the primary interface to Synomics Discover is through our automated pipeline. It follows a series of steps, each with a responsibility and purpose, all the way from user upload to the report and visualization of results. It runs automatically end-to-end to produce statistically validated genetic networks with their biological relevancy. The pipeline supports many kinds of multi-omics data in many input formats. And we will go into detail about each of these steps chronologically. And I will start to talk about the data preparation, the network discovery, and the statistical validation. And then I'll hand it over to Ryan to discuss the biological validation and output and demonstrate with some case studies. So the Synomics Discover pipeline has many entry points, depending on the data to be analyzed. And we allow for many of the industry standard data formats to be uploaded and ran. So for genomics, this includes the raw reads, the variant calls, or standard blink file formats. And for other kinds of multi-omics data, so for example, gene expression or metagenomics, we allow for read counts and proportions. So Synomics Discover is not restricted to analyze a single data source, but we're able to combine multiple kinds of omics and environmental data of interest. The quality control of this data is automated and configurable. So for example, for the minor allele frequency, the call accuracy or outliers. But it's not a requirement that the user of this pipeline knows or wants to specify these configurations. And Discover has built in sensible defaults to consistently produce applicable quality control. And likewise, Discover allows the user to specify the exact covariates, including population structure, to control for it to detect the true genetic signal. So this allows all in all for fast and reliable and accountable quality control, no matter the maturity of the customer's existing omics pipeline. So one of the core features of Discover's is our proprietary combinatorial analysis software, which Hatja also talked about. So we're combining not only pairwise features such as SNPs, but we consider multiple deep combinations of them. In each of these networks we produce, segregate the population into two groups, explaining variation in the phenotype. But the size of this multidimensional search space is huge, and this specialized software is capable of finding unique, non-redundant signaling data. This allows us to capture explanatory variables whose signal is too weak and dependent to detect using standard methodologies on the marginal distribution, for example by GWAS. So furthermore, our underlying models, they do not assume linearly additive signal genomic data, allowing epistasis and dominance effects to be detected. So likewise, it does not assume, for example, a strict ordering of higher gene expression correlating with higher yield or disease risk, but is capable of detecting intermediate relevant signals and their effect in relationship to other expressed genes. So while Synomics Discover presents these new methodologies and proprietary software, we continue to follow the best practices in data science, statistics. So by default, we split the data into three parts. The training data allows us to select from this huge potential number of networks down to relatively few, many orders of magnitude smaller. This with regularization and redundancy clustering. Of the found networks, we use a validation set to rank and filter these networks. And then on a test set, which is randomly chosen or user supplied, we can evaluate independently the performance of the individual networks. And importantly, the ranking we see of the networks based on the validation set, it corresponds well to the ranking on the test set. And that means that we're confident that the top X networks selected using this validation set from the pipeline are truly some of the best performing individual networks on unseen. And likewise, we've seen that the final estimated test set effect size, it holds well for new additional unseen data and later applications. And this means that we produce an ordered list of independently important networks, but we also produce a list just of a few feature selected networks. So for example, in the hundreds. So this feature selection such that they, when combined together as input features in some model, explain much phenotypic variation together. So we can control for the covariates directly in a user-supplied linear logistic model or similar. And these covariates could include unchangeable fixed effects and, let's say, genetic population size or mixture, such as age, season, etc. And our additional feature selection procedure uses the validation set to detect networks that in combination explain a large amount of phenotypic variance. And these could be combinations of genomic networks or even networks of different kinds of multi-omics, managerial and environmental exposure observations. And very importantly, these networks can easily be applied to new samples as they're reported as simple combinations of features. So for each of these individual networks, we produce the p-value of the coefficient by treating the network as a 0-1 factor, and the overall performance of the model, including the covariates and network by cross-validation. So these provides, for example, measures like the mean squared area or the area under curve of the model, including the network, and are compared to a non-genetic model, including just covariates. And these feature-selected networks, in the end, induces fewer needed genomic observations, which can make the data collection much cheaper. So let's say chip versus whole genome sequence. And we've also seen that with 50 networks, just 50 networks of between 1 to 5 genetic SNPs, we observe much better predicted performance of diseases such as lameness in dairy cattle compared to the industry standard models that use large amount of independent genetic SNPs. So after this introduction or to the statistical validation, I'll hand it over to Ryan to go into details about the biological validation alongside case studies.
Thanks, Tobias. Hello, my name is Ryan Morrison. I'm a senior bioinformatician at Synomics. I have a background in genetics and bioinformatics. I also have experience in molecular biology and plant science. I'm now going to go through the biological insights that Discover can generate. So now we have statistically validated combinations of features. The question is, how can Discover convert these results into interesting biology for the customer? Each output network from Discover is made up of individual features that can each provide value. To make use of this, Discover automatically produces a summary list of these network features. For genetic analysis, this could be, for example, a list of significant network SNPs. This list also contains additional useful information about each SNP, such as its position in the genome and allele information. It is then supplied in embed format, which is a format commonly used for downstream analysis using well-established genetic programs. Discover can also produce a list of genes that map to these significant network features. This is specified through user-defined parameters relating to distance. This resulting list also contains additional positional and mapping information for each gene. Discover can also automatically annotate these genes further, doing a deep dive into the biology using external databases such as Ensemble and UniProt. All this information provides the customer with a comprehensive understanding of the biology around Discover's novel targets. Discover is also able to elucidate biological pathways that play a role in the trait or disease of interest. It does this using two types of analyses. Firstly, Discover takes mapped genes discussed in the previous slide and performs a gene ontology enrichment analysis. This method highlights the functional characteristics enriched in the output set of genes, giving further biological knowledge to the customer about the potential pathways of interest in the trait or disease. The second method is to combine output networks by their shared features. This creates clusters of networks that you can see here on the right that can highlight stratification of the trait or disease response. This gives the customer biological validation and a well-defined focus for future work. This well-defined focus can then be used for a variety of different purposes, as we can see here. So firstly, in the top left, our platform can find novel discoveries in already well-researched diseases or traits. This could be used to find, for example, new gene editing targets or loss of function variants. It can also filter discoveries, so providing more focused results for challenging traits or diseases with strong signal in a particular area. It can also provide stratified targets for treatment and preventative medicine in diseases or traits with multiple causes, and provides better genomic selection for breeding purposes using the novel discoveries mentioned above. And lastly, we can see that it creates better and cheaper diagnostics based on these Discover networks. An example of this could be to reduce the number of SNPs needed to create a SNP chip panel while retaining the original accuracy of the previous panels. So I'm now going to talk about some examples where we've used Discover to provide biological insight. So we've used it to find novel methane production discoveries in dairy cattle microbiome, to improve diagnostics for mastitis resistance in dairy cattle, filter discoveries for new research in cassava mosaic disease, and to find novel discoveries relating to egg weight in laying hens. So the first example of our use was looking into how the microbiome of dairy cattle is linked to methane production is a great example of how Discover can analyze a trait using a combination of different biological inputs. In this case, it was genomic data, N16S, rumen microbiome data. This was performed by firstly combining microbiome data with the methane production data, and then doing the same with the genetic data and the trait. We were then able to determine the correlations between the trait, genome, and rumen microbiome data, and present the results here. Using standard industry analyses such as LEFC and ANCOM, there was a limited number of bacteria taxa relating to this trait. And this limits the future potential of these commercial results. However, Discover was able to identify many more novel bacteria taxa relating to methane production, and these were also linked to a difference in the underlying genetics. We've also investigated feed efficiency using this data set and found similar results. And these results, the combination of these results, should provide a customer with a set of valuable candidate targets for methane production in the microbiome that could not just be found by existing tools. Another example of us using Discover relates to mastitis resistance in dairy cattle. So mastitis is a complicated disease, and with this smaller data set we had and the low incidence of mastitis, no SNPs were able to survive the genome-wide significance threshold using standard GWAS tools. However, using only 50,000 SNPs, Discover was able to highlight 142 SNPs taken from 52 networks relating to mastitis incidence in dairy cattle. These results were found to have 58% accuracy in terms of AUC on an unseen test data set, which is a positive improvement in the predictive performance of mastitis, especially using such a low number of SNPs. Further literature search of these Discover output genes also validated the biological relevance of these results, as we discovered genes involved in pathways relating to immune response in the mammary gland, among other similar finds. Therefore, it's our conclusion that these results would provide novel targets for future diagnostic SNP panels that could not be found using standard GWAS. So in this next case study, we used a public cassava dataset looking at cassava mosaic disease incidence. This also highlights the fact that Discover can work with crops as well as previously mentioned livestock data. So previous studies on this dataset had shown a strong signal on chromosome 12 using GWAS, so strong in fact that there was barely any signal shown elsewhere in the genome. This result limits the potential opportunities for selective breeding in this species in particular. However, using 3,500 samples and 100,000 SNPs, Discover was able to find non-chromosome 12 signals of importance and also find the signal previously found on chromosome 12 by GWAS. This resulted in 274 unique networks relating to cassava mosaic disease exposure and 46 unique candidate genes. This list of networks, SNPs, and genes can therefore provide unique opportunities not found using other widely used technologies. In this last example for this presentation of using Discover, we were using it to find novel discoveries relating to egg weight in poultry. So with this project, we were able to use as little as just over 1,000 samples and 50,000 SNPs. And from that, we were able to distinguish genetic networks in poultry that related to egg weight at 56 weeks. This may not be a surprise, as standard GWAS also picks up a lot of signal. However, we found that the genes taken from our Discover platform showed more biologically relevant results after gene ontology enrichment analysis than those taken from a standard GWAS. For example, Discover genes were enriched in ontologies relating to cell development, cell differentiation, and several microRNAs found in the literature relating to egg weight in poultry. Again, Discover was able to highlight biologically relevant and novel targets not achievable using standard technologies. And now I'm going to hand you back to Haja for a summary of what we've just presented today.
Thanks, Ryan. Yeah, so in summary, wrapping it up, we've had three parts to this presentation, and I laid the foundation for why do we need a tool like Synomics Discover and the rationale behind it. That is the mountains of not only genomic, but multi-omics data, and a complex and more in-depth phenotyping in tens of thousands of individuals. So there's a big challenges. Toby has went through the Synomics Discover, the computational construct, the pipeline, and Ryan went through bioinformatics and post-bioinformatics and case studies. And what you have seen here is that we are able to provide statistically and biologically validated insights that can be extremely difficult and to some extent cannot be found using current best-in-class technologies when it comes to genome discovery as well as multi-omic data integration and discovery. It has been applied successfully across multiple livestock, crop, and other species. And in every case, we have found it very highly relevant biologically and statistically, and in most instances from novel signals. And this platform, Synomics Discover, is currently available, and we'll be really happy to discuss it further with potential customers and partners. So you have this information here, how to contact us at info@synomics.ai. So yeah, please do reach out, and I hope you enjoyed this presentation.
The genetic architecture underlying the health, productivity, and sustainability of plants and animals is turning out to be more elaborate than we might have guessed. New technologies enable us to produce and store mountains of biological data. But so far, we have only scratched the surface of what it all means. Now Synomics offers the opportunity to unlock the biological insights needed to make the discoveries that were once promised. By bringing together a proprietary combinatorial analytics and prediction engine with an industry-leading team of practitioners, we are capable of unlocking more insight than anyone else. Our platform works by rapidly identifying key combinations of features from billions of possibilities across any type of biological data set, enabling our customers and partners to quickly unlock novel discoveries. So far, we have used our platform to identify novel targets, filter discovery pipelines, develop diagnostics, and improve breeding selection. Get in touch and let us find out how we can help you unlock the next wave of biological insights.